The overall goal of this experimental procedure is the generation of infectious recombinant Zika virus from a full-length cDNA clone, assembled in a bacterial artificial chromosome under the control of the human cytomegalovirus immediately early promoter. The development of reverse genetic systems for RNA viruses such as Zika virus allow the direct manipulation of the virus genome to study multiple aspects of the viral biology and pathogenesis, and to develop vaccine strategies.
The main advantage of this technique is that the construction of Zika virus infectious clone as a bacterial artificial chromosome overcomes the toxicity associated with the fly virus genome and allows the rescue of infectious virus by direct transfection of the BAC cDNA clone into susceptible cells. This methodology could be applied to constructing stable and fully functional infectious cDNA clones for other fly viruses as well as other positive stranded RNA viruses.
To begin this procedure, construct a Zika virus cDNA clone as outlined in the text protocol, using appropriated restriction sites for DNA fragments spanning the Zika virus genome, flanked by the human CNV promoter at the 5' end and the hepatitis delta virus ribosome, and the bovine growth hormone termination and polyadenylation sequences at the 3' end are assembled in a bacterial artificial chromosome plasmid. First, grow a single colony of E. coli DH10B carrying the BAC Zika virus infectious clone in 5 ml of LB medium containing chloramphenicol for 8 hours at 37 degrees Celsius with gentle shaking.